How did Robert Koch discover tuberculosis bacteria? Imagine a time when the cause of tuberculosis was a mystery, and scientists struggled to find a clear answer. In 1882, Robert Koch changed that forever by uncovering the bacteria responsible for this deadly disease. His discovery was a mix of careful observation, innovative techniques, and scientific persistence. Koch started by developing a special staining method that used strong alkaline solutions. This technique made the tiny rod-shaped bacteria, known as Mycobacterium tuberculosis, visible under a microscope. Before this, it was nearly impossible to see the bacteria clearly in tissue samples from patients. The staining was a breakthrough because it allowed scientists to distinguish the bacteria from other tissue debris. Koch then examined tissue samples from many patients with tuberculosis. He consistently found the bacteria present in these samples. He noticed that, as the disease progressed, the number of bacteria increased, and as patients recovered, fewer bacteria were seen. This pattern helped him understand that the bacteria were linked to the disease itself. But finding the bacteria was only part of the puzzle. Koch wanted to prove that these bacteria actually caused tuberculosis. To do this, he grew the bacteria outside the body in a laboratory setting. He experimented with different nutrient media and finally succeeded in cultivating the bacteria on coagulated blood serum. It took weeks because Mycobacterium tuberculosis grows very slowly. Once he had a pure culture of the bacteria, he tested whether it could cause disease. Koch inoculated guinea pigs with the cultured bacteria, and these animals developed tuberculosis symptoms just like humans. This was a key step in proving the bacteria caused the disease, fulfilling what are now called Koch's postulates. Koch presented his findings on March 24, 1882, to the Berlin Physiological Society, marking a milestone in medical science. His work laid the foundation for modern bacteriology and helped establish that tuberculosis is caused by an infectious agent, not a hereditary condition. His techniques, staining, microscopy, cultivation, and experimental infection, are still fundamental in diagnosing tuberculosis today. In fact, the principles behind Koch's discovery are used in sputum smear microscopy, a common diagnostic test for tuberculosis. In this procedure, a patient's respiratory secretions are stained using similar methods, and under a microscope, the bacteria can be seen if present. This allows for quick and accurate diagnosis, which is crucial for treatment and controlling the spread of the disease. Koch's discovery was so important that it earned him the Nobel Prize in Physiology or Medicine in 1905. His work transformed how we understand tuberculosis, shifting it from a mysterious illness to a preventable and treatable infectious disease. Always remember, if you or someone you know is concerned about tuberculosis, it's best to consult healthcare professionals for proper testing and diagnosis.